A very good day to everyone. So today we are going to talk about RFLP and rapid molecular markers. First of all, let's see what is a marker. Well, a marker is basically anything that can distinguish one individual from another. And it is also an object that is used to indicate a position, place or route. Now, let's see what is a molecular marker. Well, a molecular marker can be defined as a fragment of DNA and it can reveal mutations. And it is also can be used to detect polymorphisms between different genotypes or alleles of a gene for a particular sequence of DNA in a gene pool. So, there are several types of markers. So, in here we are going to discuss about RFLP marker and rapid molecular marker. Now let's see about the RFLP molecular marker. Well, RFLP means restriction fragment length polymorphism. It is a restriction enzyme based approach at specific points and the resulting fragments then radio-labeled by hybridization and then they are visualized using autoradiogram. And many RFLP systems are based on change in a single nucleotide. And this marker is also a codominant marker. Now let's see the principle of RFLP marker. Well, it is a restriction enzyme digestion at specific sites that can cleave at different locations of different nucleotides. And this is based on the sequence patterns of the genome and it can thereby identify polymorphism. And then let's have a look at the procedure of how RFLP analysis is done. Well, first of all, restriction digestion of the DNA is done by using appropriate restriction enzyme. And this can be vary with the work we needed. And then we can do a gel electrophoresis in an agarose or polyacrylamide gel. And then we have to denature the DNA by soaking it in up about 0.5 m sodium hydroxide, which would separate double-stranded DNA into single-stranded DNA. And this ensures only single-stranded DNA can transfer. Next up, denatured DNA are transferred to a nitrocellulose membrane. This can be obtained by using Southern blotting technique. Then again, we have to do the baking. This has to be done with the treating with UV light. This ensures the cross-linking the DNA to the membrane. And then the pre-hybridization is done to block non-specific sites and it ensures that the probe is not binding anywhere on the membrane. Hybridization is the next step where the DNA are hybridized with the desired probe. And finally, results can be observed by autoradiogram. Now let's see how this analysis works. Assume that there's a child named Tom who lives with his mom, Jenny, and her husband, Bill. Well, one day, Jack, who is the former husband of Jenny, came and wanted to find out the biological father of Tom. In order to finding out that, DNA fingerprinting by RFLP was used. First, they have collected DNA samples from each of them In here, 3 RFLP loci were tested by using restriction enzyme EcoR1. So this restriction enzyme clears at specific sites at each individual and produce different size fragments. Then these fragments were used for gel electrophoresis. So there, different banding patterns were created. After denaturating these DNA, then they were transferred into a nitrocellulose membrane. Then this DNA in the nitrocellulose membrane were hybridized with desired radiolabeled probes, resulting fluorescence in a way that can be identified in an autoradiogram. And then these labeled DNA fragments were observed by using autoradiogram. Then according to the banding patterns observed there, results can be interpreted. As you can see, there are 2 similar banding patterns between Tom and Bill. Out of the 3 RFLP loci tested, 2 of them were same in both Bill and Tom. And therefore, we can say that Jack is not the father of the Tom. And therefore, it can be said that Bill is the father of Tom. Furthermore, to ensure these results, many RFLP loci can be tested other than 3 RFLP loci. So, I think now you have a good understanding about the RFLP analysis